Yeah, so hi everyone. It's a pleasure to be here. I always look forward for Bayesian, one of my favorite conferences. So thank you very much for organizing it. And I'm a computational biologist. My name is Nikolay Oskolkov. I'm what is called bioinformatician, and I'm employed by Lund University Biology Department. So I'm sitting in this building, I'm also employed by Scilab Lab. So I'm a Scilab Lab bioinformatician. If you know the sequencing facilities in Stockholm and today I was going to talk about our experience. I just wanted to share our experience applying Bayesian methods for integrating heterogeneous biological data. So we in computational biology, we are very pragmatic. We always think about applications. So now it's going to be a lot more about applications rather than going into Bayesian stats in depth. But I'll be talking about what kind of advantages we can get from applying Bayesian stats for real-world heterogeneous biological data. So it's about Bayesian integration of biological data, and what is well, this project started like a long time ago in our group. We started thinking about it some time ago, 2015, 2016. When people in many labs in Sweden realized that we have plenty of different types of biological data, what is called omics. So omics is a slang. Omics sort of comes from this terminology: genomics, epigenomics, transcriptomics, proteomics. So it's a multiple types of sequencing data. So when you read the sequence, biological sequence, and then quantify abundance of protein or maybe gene expression, this is what is called omics. And you can measure this on different samples, biological samples, cells, tissues. So at that time, 2015, when we started talking about this, putting this different types of biological information together and considering them in a context of each other. No one actually knew how to define it, how to evaluate it, what to expect. It was some vague idea that maybe there is some synergy between different omics that we can use. But since then, I think we grew quite a lot in our understanding. We even started teaching a course about data integration, biological data integration, and the next round will be actually in February. So if you're interested, you're very welcome to join the course. Just very quickly, what we have learned since then and how Bayesian stats can be useful here. Since I'm a computational biology, I'm a sort of an analyst. So I'm thinking about data as multidimensional arrays. Or matrices in the simplest, in the simplest form, so this is how a typical biological data matrix looks like. So it typically has two dimensions, dimension n and dimension p. So you have n columns, p rows, and n is a dimension of statistical observations. It can be samples, it can be cells. It's statistical observations. And then the p dimension is a dimension of features, what I call features. It can be genes, proteins, microbes, pixels, if you are working with image data. And then you measure each variable, gene, for example, protein abundance for many different cells. And then you quantify gene expression, for example, and this is how you get this metric. So that's our typical data in computational biology. So now, this can be sort of intuitively and graphically displayed as having n data points in p-dimensional space. And I think pretty much every statistician will tell you that in order to have a robust statistical analysis, you would really like to have n much greater than p. You would like to actually sample this p-dimensional space really well so that there are no empty corners. And then you need a lot of n. You need a lot of samples. So n should be ideally greater than p. Unfortunately, we have to live with the opposite limit. In our data, in computational biology, you very often work in the opposite limit, which is a high dimensional limit. So our data is high dimensional, and there is a lack of data. You can think about this two ways. So either you have very little data in computational biology, or you have a lot, but they are so high dimensional that you have to deal with this unfortunate limit. And this completely shapes what kind of statistical analysis we are using. So sort of depending on how much data you have, you might choose roughly one of these three analyses. And if you have a lot of data, if your N is much greater than P, you maybe want to do machine learning. If you have very little data, you better use priors that compensate for the lack of data. So in our eyes, sort of Bayesian stats, they're good because they make your sort of analysis more robust, more stable, because if you don't have data, you have to kind of invent the data, and this is what priors kind of help you to do. And I would say in biology and biomedicine is always sitting in this limit. So we have very high dimensional data. Doesn't mean that we always use Bayesian statistics. We almost never use Bayesian statistics, unfortunately. Like biomedical people, like the deeper into medicine you go, the more people use p-values and frequency statistics. And sometimes they even try to run a machine learning if they have plenty of samples. Sometimes they do. So other biology sort of areas, they're typically dealing with this p much greater than n limit, and they fall. So if you sort of try to naively run, p-value based analysis or machine learning having this limit, this high dimensional data, you run into multiple troubles, sort of singularities, divergences, etc. The curves of dimensionality. So fortunately, there is an area in life sciences which is called single cell genomics, where people recently developed technologies that allow you to get multiple types of biological information from individual cells. And since you can sort of recruit millions of cells, it becomes more and more as a routine to work with millions of statistical observations. So one cell is one statistical observation. So it's a paradise. And this is this is the area where all these integrative methods, biological integrative methods, they're blooming, making advances. And Nature even announced in 2019 single cell multi-omics as a method of the year. So this is really where we can realize all our ideas about sort of combining different heterogeneous biological data. And very, very briefly, what exactly I mean by data integration? What's the goal? What we want to get from data integration? So it can be sort of the idea, the intuition behind it is that we want to see something novel, something that is not present in individual data types. But once we combine them, we should see something novel. That's the benefit. And so graphically, it can be illustrated in the following way. So imagine that one point here is one statistical observation. And those observations, they belong to three classes: black, green, and red. Now, imagine that for each sample, for each statistical observation, you measure certain variables. Maybe it can be gene expression information or epigenetic information, methylation. It's some methyl groups on a DNA molecule that do something with gene expression. And so you can sort of imagine now that you construct a latent variable from first data type. For example, you do PCA on gene expression only, and you take PC1 and plot it as x-axis. Now, you do PCA on methylation data, you take PC1 and plot it as y-axis. So now try to project all these data points on the x-axis. You will see that the points are well intermixed. So you can't really distinguish between different classes using just one single omic. And the same is true if you project all these data points to the y-axis. You can't see difference between the classes. However, if you plot these two latent variables against each other, you suddenly can separate the three classes pretty well using just a linear hyperplane. That's the beauty of data integration, to see some patterns that were hidden in individual omics. So that's briefly the intuition. There are plenty of methods developed in this area. They all iterate around the same idea, that you should have you kind of have to find something novel after combining the data. And briefly, just to give you a flavor, what kind of techniques people use in this area. So it's a lot about graphs, a lot about networks, So for example, neural nets, they're good at sort of combining different types of data. And the idea is that you can convert the raw data into graph space. So you represent your raw data using only pairwise connections between your data points. And then in this graph space, you can intersect the graphs. You can use graph intersect technique in order to combine them. For example, this is what UMAP can do very, very well if you're familiar with this beautiful method. So at the first stage of UMAP, you construct a high-dimensional graph. So if you construct it on multiple omics, you can safely overlap them and get a consensus graph. So that's pretty much the idea. So what I call convert to common space. There are also good old ones, matrix factorization methods, such as p-less partial least square regression, canonical correlation analysis. So the idea there is that you kind of extract common variation from multiple data types and factorize it. You split it into independent components in order to sort of interpret and understand the contribution of each component to the variation. And also there are sort of Bayesian methods developed there. For example, Bayesian networks that have been already mentioned a few times at this conference. So Bayes rule, if you think, and also joint probabilities, it's kind of a natural way to put different types of information together. So Bayesian methods are also sort of quite popular in this field. And particularly, I'm going to talk about one method which became very popular, and it belongs to a family. So the method is called MOFA, which stands for multi-omics factor analysis. But it belongs to a family of methods, which I call Bayesian factor analysis methods. So they are really somewhere in between. So essentially it's a matrix factorization method, but they also kind of you can do matrix factorization in a Bayesian fashion, and then it becomes like a Bayesian factor analysis. And I sort of briefly explain how this MOFA is used, and just give you an example, practical application. How can we use this Bayesian factor analysis for real-world project and computational biology? So what's factor analysis? The major assumption of factor analysis is that there are some hidden latent variables that might capture, for example, cell type or cell site, and it's when cell divides, and this is how the morphology changes. And those hidden factors, they generate observed data, 3 different data sets: gene expression, maybe methylation, maybe mutations. So what we want to do is that we want to use this observed data in order to learn those hidden latent variables. And this is what MOFA is doing. Basically, if you have different biological data types, metabolomics, genomics, maybe proteomics, you can literally represent each of those original matrices as a product of 2 other matrices, where the Z matrix is a common matrix that captures sort of common axis of variation across all three omics, while W matrices, they're kind of loading metrics. They serve to reconstruct the original data set, and they also give you interpretation. So what's cool about MOFA is that we can get this kind of beautiful heat map. You can check, actually, using this heat map, you can check how much each individual data set contributes to these common latent factors, which is kind of a beautiful idea. You can check whether one first latent factor is driven by solely one omic, or maybe it's a combination of omics. And also, since MOFA is essentially a Bayesian framework, so it has this natural ability to deal with missing values, which is a big problem in general in data science and in computational biology as well. So we rarely have a complete overlap between different omics. Usually they are semi-overlapping. That's why missing value problem is a big headache for us. So, but thanks to this Bayesian framework, we can be quite insensitive to missing values. This is just because this community is a little bit like people like mathematics. I just wanted to show a few equations from the original paper. So it's a matrix factorization. So here you represent your original data Y as a product of two other matrices. And then you can reformulate it in a probabilistic framework. So you can define the likelihood of observing your data Y, providing these parameters Z and W. So that's your model. And you can also put priors on your parameters. And algorithmically, technically, what you do then is that you are running a variational base. Basically, this is how technically, with ELBO, this is what technically Morpho does. And now, very quickly, I show you a concrete example. What I used for my project. I had a collaboration with Wolf Reich from Cambridge, and they developed a beautiful single cell technique, which is called SCNMT. It's a technology that delivers multiple, three types of biological information, which is gene expression, methylation, and chromatin accessibility information from the same biological cells. So basically, this is how data distribution look like. They're pretty different, so it would be quite naive to just concatenate these different data matrices. They are combining binary data essentially with continuous data, which might not be that trivial. What you get, so the outcome, if you run Morpho on this SCNMT project, what you get is this very interesting understanding you can get from this heat map. What you see here is how much each biological data set contributes to each of the hidden latent factors. And the color here, it's R squared, it's explained variation. Because you can if you have two matrices, Y and X, you can compute how much of variation in Y is explained by X if you fit a linear model, and then predict Y, and then this is your model, the Z and W, that's your model of prediction. And then you can compute how much of the variation, total variation, is explained by each omic in each latent factor. And then you can get interpretation, because it's a linear algebra. You can always get loadings, you can always get ranking of your genes or CpG sites. Yes, that's absolutely the last slide I'm showing. So the most important result of Morpho is that you can get better clustering of cell types. So what you see here is a consensus plot after integration. So that's not a dimension reduction plot for each individual omic. That's an overlap of three omics, and you would not get this beautiful separation of different clusters if you use gene expression alone, for example. That's the beauty of data integration, to see something else when you add epigenetic. So that's pretty much all I wanted to say. And that's our group, NBIS, the National Bioinformatics Infrastructure Sweden. Thank you very much for your attention.